All right, so chapter 23 is called The Evolution of Populations, and the reason that this picture of the finch is here is because we're trying to remind you that a population is the smallest unit of evolution, and what that means is individuals cannot evolve. Individuals can adapt to an environment, individuals can pass their genes on to the next generation, but an individual is not evolving. So we measure evolution in terms of populations, which I would remind you from chapter 1, when we talked about all the different parts of biology and how it's organized. A population is a group of organisms, usually a species, living at the same place at the same time. So in order for evolution to happen, in order for population to evolve, we need to have genetic variation. And I made a list there, just like your book did, about defining genetic variation. So that would be alleles are involved in that. How much of the gene is expressed in a population, so in a particular, how many of the individuals in a population are heterozygous and thus express gene variability. And the third aspect of genetic variation, of course, is nucleotide variability, and that is at the molecular level, right? And your book talks specifically about introns, so I'll put this picture up here. And I'm reminding you back from chapter 17, I think it was, right? So there can be base pair substitutions, they can make changes, but depending on where those changes are, if they occur in the introns, they may not have an immediate effect on the population. All right, so the main sources of, I'll just put this up here for now. The main sources, we'll go back to me. Or actually, how about we do a share that includes a whiteboard so that I can write a little bit. Or type a little bit, right? So sources of genetic variation are important. So there are several, and I would again remind you, there are 4 that we list out in your book. The first one is that appearance of a new allele, and let me just get a text box here, this is ridiculous. All right, a new allele, which by point mutation, again, think back to chapter 17, and I would remind you here that these new alleles, when they occur, if it's a base substitution, it's not a nonsense mutation or frame shift mutation that causes death. They have the most impact on a population when those new alleles appear in germline cells. And what we mean by that is cells that will undergo meiosis, not just meiosis, right? Because the ones that are going to undergo meiosis then will be able to pass that trait on to the next generation. Secondly, second source of of genetic variation is changes in the gene number or their position. And this is something we haven't talked a lot about. We didn't speak about this particularly in chapter 17, but genes can be duplicated. And by or position, we mean if there's been crossing over, sometimes the gene cassette, the order of the genes in a particular area of a chromosome gets altered. Come to genetics, learn more about that. I have written down rapid reproduction, and this is important because in plants and animals, new genes occur about 1 in 100,000 genes per generation, so that's relatively infrequently. But if we're talking about a bacteria or a prokaryote, for example, that mutations occur about 1 in 1,000 genes, and for them a generation or generation time, we call it in microbiology, can be as short as 20 minutes. So because of the rapid reproduction, remember yesterday I mentioned that insects and birds and fish are often going to be the subject of some of these evolutionary studies that we're going to see in this chapter because they also have rapid reproduction for considering for animals. And lastly, but not leastly, certainly, sexual reproduction enhances genetic variation immensely and in 3 particular ways. Because of crossing over and meiosis, 1, prophase of meiosis 1, because of independent assortment, remember Mendel's second law, that the chromosome 2 from your mother doesn't have to stay with chromosome 2 from your father when it gets into your gametes. They can be swapped. You could have chromosome 2 from your mom and chromosome 4 from your dad and pass those on to your children. And the fact that for sexual reproduction, we're talking about 2 cells that are haploid, each haploid that have already undergone crossing over and independent assortment, and then they're going to fuse to form a zygote. So that zygote is going to get an increased amount of variation because it's getting characteristics from both parents. Now, how do we as scientists measure if a population is evolving? And that is by this measure. We call it the Hardy-Weinberg equilibrium. And this was established a long time ago, 1908, by 2 independent scientists. And literally, they were trying to come up with a mathematical formula that you could apply to Darwin's idea of evolution. And by the early 1900s, people were starting to call it the modern synthesis theory of evolution, because by then Mendel had been rediscovered. And so we were adding Mendel as a source of how the variation occurred in offspring to Darwin's idea that the variation was really important for evolution by natural selection. So these 2 gentlemen developed a theory, right, a mathematical principle, we would call it, and it is P plus Q equal squared. I don't know if I can do that. I can make that go up on this, but that's P plus Q squared equals 1, where P is equal to 1 allele for a trait or a character, and Q is equal to the other allele for that same trait. Now, if we think about expanding this formula, P plus Q squared equals 1, if you remember your algebra, it would expand to P squared, and I'm going to just draw that because I can't have it like that. P squared plus 2PQ plus Q squared equals 1, or 100% of the alleles in the population. So the Hardy-Weinberg equilibrium assumes that someone's going to count the frequency, we call it the frequency of the appearance of an allele in the population. So this p-value, you can think of this, if you remember back to our Punnett squares, if you think about a heterozygous individual for a trait, and let's just use I'm not going to use purple this time, but let's use our P characteristics of tall and dwarf. And if we had 2 heterozygotes, right? So we would end up with capital T, which actually represents the P squared, 1 character for an allele or trait. We would have 2 PQ, and we would have 1 Q squared. I'm trying to draw those as little t's, right? So these, let me change my color here. These, in a crossing of 2 heterozygotes, this would refer to P squared. We would have 2 PQ and 1 Q squared. Now, I am not going to ask you to mathematically solve Hardy-Weinberg equilibriums, but when you read through the chapter and you see their examples where they score these as percentages, right? And the reason it's scored as a percentage because it means how many of the total of the population, that 1 or 100 represents how many of those alleles in the population. And the percent is the frequency of P in that population. Again, I'm not going to ask you to solve Hardy-Weinberg equilibriums, but I do want you to have an understanding of how they work. Now, Why do we care? Why would anyone want to count allelic frequencies? Trust me, people do. Why would they want to do that? Because the purpose of the Hardy-Weinberg equilibrium is to check or monitor if a particular population is evolving. It's a measure of an evolving population. Now, because it's a mathematical principle, it has conditions. And I'm not going to retype them, but there are 5. And you can see them. They're in a table in your book, all spelled out for you. Table 23, 1. And they're listed there, and I'm just going to say them out loud. The population should have no mutations. All the mating should be random. There should be no obvious natural selection. The population needs to be extremely large because we're using a mathematical calculation here, so we need to like flip that coin 100 times, not 10. And there should be no gene flow, which means reproductive individuals need to be isolated from leaving, we would call that emigration, or new alleles coming into the gene pool, immigration. Okay, now you can clearly see that you can't meet all of these criteria, right? There's no way to meet all of these criteria. However, you want to limit them. If you're studying evolution, you would want to limit as many of those variables as possible and keep as many of those conditions at minimum as possible. So this is, of course, presents a little bit of a difficulty because you would study the population. Usually people study the population out in nature, and then they recreate some aspect of that population inside their laboratory, right? Or in a, maybe if it's a plant they're studying, maybe they would recreate it in a particular field where they can kind of isolate more of these variables to study the evolution, or not. Maybe they're just going to study it in the wild. So your textbook is going to focus on 3 of those 5 from that list that cause the most changes to the frequencies of alleles in the gene pool. And so those are the 3 that we're going to talk about. The first one's genetic drift, second gene flow, and third natural selection. So I'm going to go back to sharing my screen. Let's take a look at this. So this is just a picture of the Hardy-Weinberg equilibrium again at to show you a demonstration of the population of alleles, percentage of alleles in a population. So we have the red allele here and the white allele. And I believe the example that we're focusing on is, remember our flowers that had incomplete dominance. This is why we bothered to teach you that predominantly, because that incomplete dominant allele, the flowers that have 1 gene for red and 1 gene for white, they are the 2pq, right? And they're very easy to see in the population. This would be the p squared, this would be the 2pq, and this would be the numbers of white. And what you can see, if you went through all the calculations, which remember, I'm not going to ask you to do this, is that the frequency of the alleles stays the same. It's relatively stable if the population is not evolving, which makes sense, right? So there's just the picture of the flowers again and the frequencies, right? And so now we're going to talk about what can happen, what we call genetic drift, which is a dramatic change. Usually it has to do with the population size being small. And so here's a bunch of flowers that are growing, let's say out in my yard, right? And I've got the p squared flowers, the red ones, a few pink ones, and 1 white one, right? And in this initial population, which, like I said, let's use my backyard, initial population, I've got a frequency of p at 0.7 or 70%, and a frequency of q, which in this case is the white allele, in a much smaller percentage, right? All right, so along comes a deer, or a turkey. I've seen both of those in my backyard. And the deer was rather unexpected, but let's say the deer comes along and it eats the flowers, Okay, I believe it's going to eat all of these flowers, and all that I have left, I see it out there eating my beautiful bouquet, and I run out and I say, get away from my flowers. And all that's left are the 2 red ones. Now, when these 2 red ones reproduce in the next generation, you can clearly see the frequency has dramatically changed. Now I have P at 100% and I have no Qs left. I have no white alleles left because it ate the pink flowers and the white flowers. So this is a demonstration of how you could use the Hardy-Weinberg equilibrium by measuring the frequency of these alleles in a small population. You can get genetic drift very quickly in 2 or 3 generations here. We've got this genetic drift from 70% and 30% to completely red at 100%. Again, I'm not going to ask you to solve Hardy-Weinberg equilibriums and tell me the percentages and the shifting of the alleles over time. You'll learn to do that later. So now, when humans are the, not the deer randomly eating flowers in my backyard, but when humans are involved and we have a small population, so think of any species that's endangered, we call that specifically the bottleneck effect. And what's meant by that is here's a original population, and let's say they have 3 alleles for a particular trait, or this is just all the alleles in this original population. And the population experiences a bottleneck, which means there are few survivors. And again, this could be because of some sort of human intervention, hunting, land development, pollution, whatever you want to think of, okay? And only a few survivors leave alleles, and notice in this surviving population, we would get a huge shift in the allelic frequencies because you can clearly see there are none of the yellow alleles make it through, and only one of the white alleles make it through, predominantly they're blue. I usually use an example of the cheetahs for this. I think I listed it probably on your handout, in that the wild population of cheetahs originally had lots of different alleles for all kinds of characteristics, let's say speed, coloration, so that they were camouflaged, eyesight, rearing their young, and that population went through a severe bottleneck, primarily due to hunting and land development, meaning humans using more of the land for food and having the cheetahs having a much smaller range. Primarily, I'm thinking about in Kenya. And then the humans were like, oh no, we need to save the cheetahs. Okay, now there's only this many alleles left. And what if this yellow allele was the one for being able to run really fast, or being able to feed your babies, or being able to be the right color pattern so that you're hidden very well, or maybe it's a characteristic that it has to do with picking your mate. You see how now in the surviving population, because that allele isn't there, this is a much weaker population. Your book also goes through the prairie chicken, the greater prairie chicken, which was alive in the 1800s and lived quite across most of the Great Plains. And then this is showing you another bottleneck effect, where in Illinois in particular, there was a lot of hunting because it's kind of the jumping off place for westward expansion. And by the 1990s, there were only 50 prairie chickens left, compared to Kansas and Nebraska, where there aren't as many people still, and there were more prairie chickens, right? You can see that originally the prairie chicken population, from historical data, which they could collect from molecular analysis of feathers and stuffed prairie chickens, taxidermied prairie chickens, as well as the live populations in these countries, in these states where there wasn't a bottleneck effect. Notice how many more of the young survive. And in the case where there's been a big bottleneck, only about half of the eggs actually hatch. This is an example. So the prairie chicken got put on the endangered species watch list because its genetic variability was so low. And so some scientists at the, I think some government department, they intervened and they took some prairie chickens that I think it ended up using them from Missouri, actually, where there had been less of a bottleneck, and they repopulated the Illinois population to see if they could increase the number of eggs that hatch, and the answer is yes. And you're like, okay, why do we need to save the prairie chicken? Well, not only just for biodiversity, but it was an example of an experiment designed to see if humans could re-intervene and undo a bottleneck effect. And this was used as the basis for trying to increase the genetic variability in the cheetahs. And so experiments have been done that have increased the gene pool in the wild cheetahs by using artificial insemination, actually, of wild cheetahs with zoo cheetahs, because the zoo cheetahs had actually not gone through the same bottleneck, and they had different alleles for their characteristics, and it's actually helped the wild cheetah population. All right, so gene flow. This is a new experiment to me. It's new for this edition of the book, and I've never used it before, but it's near and dear to my heart because this is basically where I was born and raised, near here, near Kelleys Island, this little part of Ohio. And this is Canada here, Pelee Island. This is some water snakes that live in Lake Erie, and they live in Canada and Ohio. And it turns out that this is an example of gene flow, because the snakes on these islands, particularly Middle Island here and some of the Bass Islands, they actually can swim from one island to another. And so the population actually keeps changing. There's the unbanded ones versus the banded ones, right? And you can see that on the Ohio mainland and in Canada, there's predominantly, I think this is supposed to be the banded ones, right? And the unbanded are here in the islands, but you still keep seeing banded ones occasionally or unbanded ones occasionally, because the snakes can actually move from place to place. So that'd be an example of gene flow, because they're actually immigrating and emigrating. And so we start to see banded on both the mainlands. I guess this is the other, it's bigger, I guess. All right, now the third area of the Hardy-Weinberg equilibrium that your authors are going to focus on is natural selection, because of course we keep harping on how natural selection is the main motive force for evolution. So we're going to talk about different types of natural selection, different categories of natural selection. I would probably do these in reverse order: stabilizing, disruptive, and directional. I think there are examples in your book of all of these. We're just going to look real briefly at these mice, and we're looking at mice that have various colors from very light to very dark, with some brown mice in the middle, what we would call agouti mice. Stabilizing selection is measured, or you would call a particular type of evolution or selection, subcategory of natural selection, stabilizing. If you had studied this population using the Hardy-Weinberg equilibrium, and the resulting population at the end of your study was mostly what we would call the mean, in this case, the brown mouse, right? The extremes in stabilizing selection, the extremes are selected against, maybe because of predation. Let's say there's a hawk that is a predator of these mice, and it can clearly see the white mice and has easy time seeing the very darkest mice, but the ones that blend in with the environment, it can't see, so they survive and leave the most offspring. A different type, subcategory of natural selection is what we call disruptive, and you can see the mean is being selected against here. See how the white arrow is pushing down? That means, so if this was an original population forms a bell-shaped curve, in the case of disruptive selection, the mean is selected against and the two extremes are selected for. So let's say there's some sort of incident where a road gets put in through the grass, and the mice that are closest to the beach and lightly colored can blend in, and the mice that have the darkest color can be blend in closest to the road. and that area where the brown mouse fit is no longer there, so the hawk can see them best, right? That would be an example of disruptive selection. And directional selection is over time, one of the extremes is selected against, and the mean and the other extreme is selected for. And this example could be either direction. See how the arrow is pressing down here on the white mice in this case. Directional selection could also happen the other way, where the other extreme was selected against, and the mean and this extreme was selected for. Again, that usually indicates major change in the environment. And again, there are examples in your book that you can go through and read. I don't have time to go through all of them with you. Okay, now I would add to that list sexual selection. Your book has, I guess we look at the peacock for that, and balancing selection. So I want to go through those real quickly as well. Sexual selection is usually a result of sexual dimorphism. This is the male peacock with its very fancy feathers, and this is the peahen. This is very plain, and it's like it sounds. The peahen is going to choose a mate based on the appearance, who has the longest feathers, basically, and most brightly colored feathers. I think this is another example with singing, right? So it's some frogs. And we're looking at the female choosing the male frog that has a particular song. This is an example of balancing selection. We used to call it frequency. This is particular to frequency dependent. These are some predators, fish that attack this other fish, and it's called frequency dependent because some of these predators have a curve to their mouth to the left and others to the right. And what we see over time, basically over 1 or 2 years, is we see an oscillation between the left mouth and the right mouth. And the reason for that is because the fish that's being attacked during its lifetime becomes better at getting away from, if it's a year where there's a lot of right mouth, it gets better at defending itself against right mouth attacks. So fewer of those reproduce and more of the left mouth reproduce. So the next year there's more left mouth, fewer right. So we say that the more common phenotype is selected against and the least common phenotype is selected for. And this alternates, as you can see. Yes, someone actually counted these fish and counted the alleles that contribute to being left-mouthed or right-mouthed. We used to have a picture in the book of the actual fish so you could see what the left mouth and right mouth looked like, but they don't have that in there anymore. But you can see for almost at least about 10 years, some group of scientists counted them and looked to see the frequency of left mouth individuals, and we can see the frequency is high and then low, and then when they reproduce more, leave more babies, and then when they don't, and then when it changes again. And this other piece of balancing selection is called heterozygote advantage, and the example of that is with the sickle cell trait or the sickle cell allele. And we know this if we go all the way back to chapter 5, we saw in the protein formation of hemoglobin, the difference between normal hemoglobin and sickle hemoglobin shape, and it has to do with a single base mutation, right? So a single point mutation from chapter 17, and the resulting shape change of the protein changes the shape of the red blood cell, which then causes basically some clogs in arteries, compared to the more fluid and flexible donut-shaped red blood cells. Now, why do we call this heterozygote advantage? Because that has to do with this little guy, this little mosquito, that spreads malaria. The parasite, the malaria parasite, is called Plasmodium, and the parasite survives better in organisms in red blood cells that are not sickled. So by being heterozygous and having some of your red blood cells normal shaped and some of them sickle, you actually get more protection, more innate protection from malaria. So we can see the frequency of the sickle cell allele is higher in populations where there are these mosquitoes that spread malaria, because if you keep this population heterozygous, right? So by natural selection, look, it's all in places where there are lots of mosquitoes. And they're not showing us South America because they can't show us the whole globe at once. But this is also here in parts of South America, obviously, because it's the same part of the world, right? The same latitude and longitude. So organisms, people who are homozygous for the sickle trait are not going to survive very well, but people who are heterozygous, they have an advantage because they're going to be more protected from malaria. People who are homozygous without, they're living where it's more likely that malaria is not, and so they're going to survive here. But if they move to these locations, they of course would be at a big disadvantage. So that's why we call it heterozygote advantage. This chart just lists those 5. This is a picture of table 23.1, which lists the 5 conditions of the Hardy-Weinberg equilibrium. And I think that is pretty much the end of this chapter. So stop the share, and I'm also going to stop the recording.